سلام عرض میکنم خدمت همه حضار محترم. روز دوم پنجمین همایش آرا و آثار داروین رو در خدمتتون هستیم که به مناسبت روز جهانی داروین برگزار میشه و تیتر این پنجمین همایشمون هم هستش تکامل انسان. امروز 5 تا سخنرانی خواهیم داشت که در خدمت جناب آقای جلال سلطانی، آقای مهرداد عربستانی، مارک استون کینگ، برنارد بود و کریگ استنفورد هستیم و خب روز دومه و روز سوم رو هم که فردا همین ساعت خواهیم داشت. برای کسب اطلاعات بیشتر و ویدئوی همایش‌ها و دسترسی به ویدئوی همایش‌ها به کانال‌های تلگرامی مدرسه مجازی فلسفه تردید و خبرهای تکاملی هم میتونید در واقع رجوع کنید که آدرسشون هم توی چت باکس نوشته شده. خب با توجه به اینکه برای آقای سلطانی فکر میکنم که مشکل لاگین کردن پیش اومده، اول شما درخواست کنیم. سلام.
سلام علیکم. سلام علیکم.
خیلی متشکرم. خیلی ممنون. جناب آقای دکتر مهرداد عربستانی، ایشون استاد گروه انسانشناسی دانشگاه تهران هستند و البته که ارائهشون به زبان انگلیسی خواهد بود. بنابراین خیلی ممنونیم که دعوت ما رو پذیرفتید. در خدمتتون هستیم.
Okay, thank you very much. I'm really honored to be invited to contribute in this symposium, and I'd like to express my appreciation to Mr. Memar School of Terdid. This event happened. So the title of my lecture is Intersectionality of Genes and Language. So for this, I'm going to reflect on the nexus between biological evolution and the world of language. As we delve into intersectionality of genes and language, we embark on a journey that unveils the intricate interaction between our evolutionary history and the symbolic register that defines our humanity. Without any claim of comprehensive discussion, here I only try to point out this intersectionality and its consequences through some examples. Language, a remarkable product of our biological evolution, stands as the hallmark that distinguishes us, human sapiens, from other primates. It transcends a mere tool for communication, representing a distinct dimension in our evolutionary journey, one that surpasses the conventional survival of the fittest paradigm. The homogeneous evolution over about 2 million years culminated in the appearance of Homo sapiens approximately 300,000 years ago. Since the homogeneous includes animals that live in groups in small face-to-face communities, the evolution was initially tailored for survival in these small groups. Our genetic makeup underwent a profound shift as human societies expanded with the advent of civilization around 7,500 years ago, a relatively short span of time compared to the genus' long evolutionary course. What distinguishes us from our primate kin is our enlarged and complex brains and their capability to develop a highly abstract symbolic order or language. This level of language capability marks the key divergence between humans and other primates. This linguistic evolution is the cornerstone of our uniqueness and a testament to the transformative power of language as it intertwines with our genetic destiny. While genetics lays the foundation for the human body, language significantly influences the trajectory of human evolution. Language shapes collective identities through stories that define values and behaviors that can alter the course of evolution. Next slide, please. Homo sapiens are the storytellers of the genus. Consider the narratives we weave through language. We craft stories that form collective identities, defining values and behaviors. Religion, nationality, church, or ummah are not mere tales, but powerful discourses that unite us as and determines our responses to the environment. Joseph Henry's groundbreaking work, The Weirdest People of the World, unveils how the trajectory of our evolution can deviate from the genetic blueprint through the power of stories. Henry's exploration of a seemingly simple edict, don't marry your cousin, is a captivating saga of linguistic intervention. This rule, the rule of don't marry your cousin, promulgated by unknown early church fathers, unleashed a cascade of changes, starting from replacing endogamy with exogamy, and the result will be replacing group and tribal bonds with individual-centered networks. States replaced tribes, science overtook law, and law superseded custom, a transformative journey that resulted in societies and people that were weird, according to Henrich, which is an acronym for Western, educated, industrialized, rich, and democratic. They are weird, according to Henrich, because they are different from the majority of societies in the world who are living inside other stories. Yet, the shadow of our tribal past still haunt our societies through behaviors such as ethnocentrism and nepotism. Rooted in our evolution in small communities, they could pose threats to our survival in an era marked by widespread violence and environmental crisis. While these behaviors might have had evolutionary advantages in specific contexts, it's crucial to note that human societies have evolved to be highly diverse, and cultural, legal, and ethical systems often seek to mitigate the negative aspects of ethnocentrism and nepotism. Contemporary societies strive for principles such as fairness, justice, and equal opportunity, even when these may run counter to some aspects of our evolutionary past. We have an inherent tendency to distinguish and prioritize in-group over out-group in a face-to-face small community, a tendency encoded in our genetic throughout the procedure of evolution that makes us support our kin and fellow small group members over the outgroup. However, the advent of civilization in around 7,500 years ago required new stories to integrate people into the bigger communities. Instead of stories about common ancestors that is associated with tribal identity, New stories emerge around common beliefs, common loyalty, and the common cause, or the common political entity, and so on. That is, they lay the ground for the identities like church, ummah, or nationality, or class, and even global identities. Identifying with a big community requires members who never see each other but identify themselves in an imagined community that is based on a story, surpassing the face-to-face community of small groups in early human history. Actually, these stories are the discourses and that can or enable the people to identify themselves inside these stories and identify themselves as the member of a big community, an imagined community. Ethnocentrism may once have been adaptive to human survival by reinforcing in-group commitment, solidarity, cooperation, and preserving scarce resources. In the same way, nepotism and mistrust toward strangers could have helped protect the group integrity. However, in an era of worldwide prevalence of violence, sectarianism, and ethnic and nationalist conflicts, and intolerance, ethnocentric tendencies are potentially prone to fuel animosity and threaten human well-being and survival on Earth. Nepotism may lead to familial oligarchies that do not necessarily favor the community's survival, and in most cases deteriorate the situation by depriving the society of its meritorious members. From our post-tribal and pluralist perspective, and in an era where global threats are self-evident, we can readily see the need to attenuate ethnocentrism if the whole of humanity is to survive. In other words, some human traits, as the results of human evolution in small communities, may be maladaptive today and hurtful to human society. In the face of world war crises such as global warming, violence, intolerance, and mass migration, embracing comprehensive stories that transcend tribalism is a necessary cultural and discursive intervention, a beacon that may secure our collective future on Earth. Uh, the next slide, please. Another example that I want to take here is the example of epigenetics and how language is involved in the transmission of epigenetic adaptations. Epigenetic, the genetic adaptation to environment, is a well-known phenomenon. Language and communication can potentially play a role in transferring epigenetic modification to the next generation. However, the mechanisms involved are still an active area of investigation. It is suggested that the language and communication within the family or social group can contribute to the transmission of behaviors and cultural practices influenced by epigenetics in former generation. We can take as an example the high occurrence of diabetes and obesity among South Asians that resulted in what we can call the skinny fat body, low muscle mass and accumulation of fat around the waist. Beyond lifestyle factors, Dr. Mobin Sayed, um, medical doctor and radiologist, offers a historical lens to explain the high occurrence of diabetes among South Asians. According to him, South Asians enduring 31 one famines during the British Raj in 18th and 19th century India, and they developed a starvation-adapted genetic tendency to store fat. This adaptation surprisingly transferable to the next generation and become an evolutionary mismatch in the modern era of abundance. Remarkably, this genetic adaptation transcends biology, reaching into the realm of language and culture. Acculturation, a discursive procedure, passing down beliefs, practices, lifestyle, values, and fears and worries from one generation to the next, becomes a conduit for the inheritance of psychological trauma, such as the fear of starvation and hunger, and consequently lead to the persistence of the original epigenetic changes. The fact that surviving a single famine doubles the risk of diabetes and obesity in the next generation illustrates the profound impact of language and culture on our genetically encoded fixtures. In fact, a culturally constructed situation such as colonialism causes an epigenetic effect, which in turn is transferred to the next generation through acculturation. To recap, in one sentence, language itself, a product of biology, adds a dimension to biological evolution that makes unprecedented complexities. Let us carry forward to understanding that our evolution is not solely written in our genes, but scripted in the narratives we articulate through language. In this intricate dance between our biological essence and cultural expression, we find the essence of our shared human experience. Well, thank you very much.
Yes. خیلی ممنون های دکتر عربستانی، خیلی متشکرم ازتون. فکر میکنم که آقای دکتر سلطانی الان هستند در جلسه و ما میتونیم در خدمتشون باشیم. آقای دکتر جلال سلطانی استاد گروه گیاه‌پزشکی دانشگاه بوعلی‌سینا هستند و مترجم کتاب ژن خودخواه که دعوت ما رو پذیرفتن و سخنرانیشون با عنوان تکامل آدمی از منظر ژنوم رو برای ما برگزار میکنن. آقای سلطانی، صدای من رو میشنوید؟
خب به نام خدا، ضمن عرض سلام حضور تمام حضار گرامی و اساتید محترمی که تشریف دارن و دوستان و همکاران داخل ایران و خارج از کشور که زحمت این روز رو کشیدن و بیشتر بار این برنامه به دوش دوستان بوده. بنده خوشحالم که در خدمت شما هستم و عنوان صحبتمون همین که دوست گرامیمون عرض کردم، تکامل آدمی از منظر ژنوم هست. خب ژنوم چی هست؟ ژنوم مجموعه دی ان ای هست که داخل یک سلول وجود داره و توی انسان این ژنوم دو پاره هست، یعنی بخشی از ژنوم داخل میتوکندری هست که در واقع اندامک تنفسی سلول برای خودش ژنوم داره و ژنوم اصلی که ژنومیک دی ان ای هست توی هسته سلول وجود داره و مجموعه این دوتا با هم هست که ژنوم رو پدید میاره. بنابراین ژنوم، حالا اون چیزی که توی سلول هست، هر سلول جدا از ژنوم هسته‌ای خود ژنوم‌های اندامکی هم داره و در مورد انسان هم همینطور هست. خب ما یک نگاهی بکنیم به وضعیت درخت حیات از دیدگاه زیست شناسی. همینطور که ما توی این تصویر می‌بینیم، حیات از منظر سلول شناسی به 3 گروه تقسیم میشه: باکتری‌ها، آرکیا و یوکاریا. باکتریا و آرکیا که قدیمی‌ترین موجودات کره زمین هستند، از منظر سلول شناسی تک سلولی هستند. ما بهشون پروکاریوت میگیم و ساختارهای سلولی ساده‌تری دارن و اگر توی این شاخه سمت راست این درخت فیلوژنتیک نگاه بکنیم، یوکاریوت ها مشتمل بر موجوداتی هستند که عموماً پرسلولی هستند، توشون تک سلولی هم هست ولی عموماً پرسلولی هستند و از نظر ساختار سلولی و اندامک‌های که داخل سلول‌هاشون دارن کمی پیچیده‌تر از باکتری‌ها و آرکی‌ها هستن و اگر ما بالای این شاخه رو نگاه بکنیم 3 گروه از این موجودات یوکاریوت از نظر تکاملی نسبت به بقیه پیشرفت بیشتری کردند که از جمله گروه جانوران، قارچ‌ها و گیاهان که انسان در این گروه جانوران قرار میگیره و از منظر تکامل سلولی ما تقریبا همرده گیاهان و قارچ‌ها سلول‌هامون تکامل حالا بیولوژیک پیدا کرده و جالبه که توی این درخت فیلوژنتیک آرکیا بیشتر از اینکه به باکتری‌ها نزدیک باشن به یوکاریوت‌ها نزدیکن و این پدیده‌ای بود که توی 2 دهه گذشته مشخص شد. خب ما اگر نگاه بکنیم که رابطه انسان با دیگر موجودات از جنبه تکاملی و گذر زمان در این بازی تکاملی چطور هست، من یک درخت فیلوژنی اینجا آوردم که یک سری موجودات مدل رو توی این درخت فیلوژنی ما می‌بینیم. این موجودات مدل موجوداتی هستن که توی زیست‌شناسی خیلی کاربرد دارن تو کارهای آزمایشگاهی و بیشتر تحقیقات آزمایشگاهی روی این موجودات انجام میشه. مثلا کبک لزج، مخمر نان، نماتود، مگس میوه، خارپشت دریایی، ماهی، قورباغه، مرغ خانگی و موش که در نهایت انسان. ما توی این تصویر اگه نگاه بکنیم اعدادی که روی این درخت وجود داره 90 310 و اینها در واقع بازه‌های زمانی تکاملی هستن بر مقیاس میلیون ساله. اگر نگاه بکنیم فاصله تکاملی انسان و موش در واقع نیای مشترکی که این دو موجود داشتن بیش از 90 میلیون سال ازش میگذره و حدود 90 میلیون سال پیش این دو از هم منفک شدن و همینطور به نسبت موجودات دیگر اگه نگاه می‌کنیم پایین‌تر میریم این فاصله‌های تکاملی طولانی‌تر هست و میرسه مثلا به نماتود که حدود 1 میلیارد سال فاصله تکاملی داره و به مخمر نان و کبک لزج که بیش از 1.5 میلیارد سال فاصله تکاملی هست و توی تصویر سمت چپ هم ما می‌بینیم از اون خاستگاه پروتوسل هایی که اون اوایل تکامل پدید اومدن تا بیاد توی دریاها ماهی‌ها پدید بیان، دوزیستان و مهرداران و حالا پستانداران تا برسه به انسان. توی این بازی تکاملی انسان نیای مشترک با بسیاری از موجودات داره. خب در داخل ژنوم انسان چی هست؟ ژنوم انسان ما به انسان میگیم دیپلوئید، 2 سری کروموزومی در سلول‌های ما وجود داره که ما هر سریش رو از یکی از والدینمون میگیریم، یک سری از مادر، یک سری از پدر و به اینها میگیم کروموزوم‌های هومولوگ. 2 تا کروموزوم هم هستن که بهشون کروموزوم‌های جنسی میگیم، تحت عنوان کروموزوم‌های X و Y ازشون نام میبریم که ترکیب اینها که XX باشه یا XY باشه جنسیت رو توی فرزند تعیین میکنه و مجموعه ژنومی ما یک مجموعه 2 سری هست، یعنی یک سری از پدر یک سری از مادر و ما یک سری این رو میگیم تحت عنوان ژنوم. ژنوم انسان در دهه 1990 تا سالهای 2003 طول کشید تا تولیابی بشه و در سال 2003 نسخه نهایی ژنوم انسان بیرون اومد و پرسش‌هایی توی این تولیابی پدید اومد که نیاز بود که این پرسش‌ها پاسخ داده بشه. یکی از پرسش‌ها این بود که آیا اندازه ژنوم مهم هست در تکامل موجودات؟ آیا انسان رو که ما موجود پیشرفته می‌دونیم آیا ژنومش نسبت به موجودات دیگه ژنوم بزرگتری هست؟ تحقیقات نشون داد که نه اینطور نیست. اگر ما توی این جدول نگاه بکنیم یک مقایسه‌ای هست از ژنوم موجودات یوکاریوتی که تولیابی شدن در 2 دهه گذشته و جایگاه انسان رو به عنوان هوموساپینس می‌بینیم که ژنوم انسان از نظر اندازه حدود 3235 مگابیس هست، یعنی حدود 3 میلیارد و 200 میلیون بیس هست. بیس پیر منظور اون توالیه‌های نوکلئوتیدی هست. ولی اگر نگاه بکنیم اینجا ما یک موجود تک‌سلولی آمیب می‌بینیم، آمیبا دوبیا که تصویرش رو من بالا گذاشتم. اون تصویر موجود تک‌سلولی حدود 200,000 مگابیس اندازه ژنومش هست، یعنی تقریباً 60 برابر انسان اندازه ژنوم این موجود تک‌سلولیه. یا 2 تا موجود دیگه که گیاه پاریس هست و یک نوع مارماهی توی دریاها که اندازه ژنوم اینها می‌بینیم که 165,000 مگابیس و 143,000 مگابیس هست و اگر به گیاهان نگاه کنیم مثلا توی گندم و نخود ما می‌بینیم که گندم حدود 16,500 مگابیس اندازه ژنومش هست. ملخی که حالا ما می‌بینیم توی صحراها 6,500 مگابیس اندازه ژنومش هست و گیاه نخود 4300 مگابیت. تمام اینها اندازه‌های ژنومشون از اندازه ژنوم انسان بیشتره بنابراین اندازه ژنوم یک معیاری نیست که نشون بده که پیچیدگی موجود یا تکامل موجود در چه حد هست. این اندازه ژنوم معیار درستی برای ارزیابی تکامل نیست و ما اینجا من تصویر نمادود سلام ربیتیس الگانس رو هم گذاشتم به این خاطر که از جنبه مقایسه با انسان این موجود جالبه. این موجود ژن‌هاش به اندازه ژن‌های انسان هست، ولی خب خودش میکروسکوپیه، ما اینو به چشم نمی‌بینیم، اندازه بسیار کوچیکی داره و اندازه ژنومش حدود 100 مگابیسه که تقریبا 1/30 اندازه ژنوم انسانه. بنابراین ما می‌بینیم که از نظر اندازه ژنوم توی موجودات وریشن یا تنوع بالایی هست و لزوما هر موجودی در شاخه‌های بالای رده حیات به این معنا نیست که ژنومش هم بزرگتر هست. خب پس موضوع چیه؟ سوال بعدی اینه که چه تعداد ژن توی این ژنوم‌ها وجود داره و ما آیا بر اساس تعداد ژن‌ها میتونیم تکامل رو تعریف بکنیم؟ آیا موجودات پیشرفته‌تر ژن‌های بیشتری هم دارند؟ خب یک نگاهی می‌کنیم، یک مقایسه می‌کنیم اینجا. اولا که یک تصویر من بدم اینجا خدمتتون کاریوگرام کروموزوم‌های انسان هست که نشون میده که تراکم ژنی توی
بخشایش
تک تک جانم.
شما من فکر میکنم که شیر اسکرینتون رو روی یک قسمت دیگه گذاشتید و ما فقط تصویر اصلی پاورپوینتتون رو میبینیم، اون قسمتی که در واقع
الان شیر شد یا نه؟
الان درست شد، ولی وقتی که اسلایدر رو عوض میکنید ما دیگه چیزی نمیبینیم. فکر کنم از
الان به این
همینجا
شکل
اسلایدر رو
می‌بینی؟
عوض.
الان
بله.
خب من
الان
پس اینو صفحه‌شو کامل میکنم، صفحه رو میکشونه شما نمیبینید، در حالت
بله
کامل نیست، من همینطوری پیش میرم.
بله مرسی.
خب من یک بار دیگه این تصویر رو پس خدمتتون نشون بدم که ژنوم انسان در مقایسه با موجودات دیگه ژنوم چندان بزرگی هم نیست. ما موجودات دیگه داریم مثل این تصویر بالایی آمیب هست یا گیاه پاریس هست یا موجودات دیگه‌ای که ژنوم هاشون خیلی بزرگتر از ژنوم انسان هست. بنابراین اندازه معیار مناسبی برای بررسی تکامل نیست. خب توی این تصویر ما کروموزوم ها رو می‌بینیم و تراکم ژنی روی کروموزوم های انسان، جاهایی که تیره رنگ هست نواحی هتروکروماتینی هست و نواحی ژنی جاهایی هست که تیره خاکستریه، یعنی رنگ نوک هست. اونجا جاهایی هست که تراکم ژنی توی کروموزوم ها بالاست و ما می‌بینیم که توی این نواحی بین این نواحی دیگه هم هستن که ژن توشون وجود نداره، نواحی بین ژنی هستن. خب ارزیابی دانشمندان از تعداد ژن‌ها توی انسان این بود که انسان باید بیش از 100,000 ژن داشته باشه، به این خاطر که توی سلول‌های انسان بیش از 100,000 نوع پروتئین تولید میشه. ولی ارزیابی‌هایی که صورت گرفت نشون داد که تعداد ژن‌های انسان کمتر از 20,000 تاست و این دانشمندان دچار چالش کرد که این معیار 100,000 پروتئین و 20,000 پروتئین چطوری باید حل بشه. حالا بعدها تو بیان ژن تو بحث آلترناتیو اسپلایسینگ و پردازش تناوبی این موضوع مشخص شد که هر ژن میتونه چندین نوع پروتئین تولید بکنه و همین خاطر تعداد پروتئین‌ها بیشتر از تعداد ژن‌ها هست. ولی خب تعداد ژن‌های انسان هم لزوما تعداد بالایی نیست. ژن‌های کدکننده پروتئین همونطور که می‌بینیم هولوش 20,000 تا کمتر از 20,000 تاست تا به امروز اون چیزی که مشخص شده. اگر بخوایم مقایسه بکنیم می‌بینیم که توی این جدول مقایسه کرده انسان رو با بعضی از این جانداران دیگه. مثلا مقایسه انسان ما با برنج حدود 35,000 ژن داره یا گیاه آرابیدوپسیس حدود 27,000 ژن داره یا مرغ خانگی 15,000 تا، مگس سرکه 13,000 تا و مخمر حدود 6,000 تا. و جالبه که تعداد ژن‌های نماتودی که اونجا خدمتتون عرض کردم نماتود سلول ربیتیس الگانس با انسان تقریبا یکسانه. انسان 20,000 تا ژن داره و این نماتود هم 20,000 تا. ما توی اون شاخه تکاملی گفتیم که فاصله تکاملی انسان با این نماتود نیای مشترکشون برمیگرده به 1,000,000,000 سال پیش و این نماتود یک موجود میکروسکوپی هست، به چشم دیده نمیشه، خیلی ریزه. هرچند اندازه ژنومش حدود 1/30 ژنوم انسان 100 مگابیسه ولی تعداد ژن‌هاش به اندازه تعداد ژن‌های انسانه. ما می‌بینیم که اینجا باز ما با یک تناقض مواجه میشیم. چرا ژن‌های یک موجود میکروسکوپی به اندازه ژن‌های ماست و فرق ما با این توی تکامل چی هست؟ اگر نگاهی به کاتالوگ ژن‌های انسان بکنیم که کارکرد این ژن‌ها چی هست در مقایسه با موجودات دیگه توی این تصویر، ما می‌بینیم که بیشتر ژن‌ها توی انسان در مقایسه با موجودات دیگه از نظر فعالیت‌های پرهم کنشی، یعنی پرهم کنش پروتئین ها و پروتئین با دی ان ای و آر ان ای با دی ان ای، انسان واجد تعداد بیشتری از این ژن هایی هست که برهم کنش میدن در واقع پروتئین هاشون با ژن های دیگه. و توی این تصویر هم ما می‌بینیم که اومده اینها رو به تفکیک مشخص کرده که من حالا علامت گذاشتم که به نظر میرسه که توی ژن‌های انسان پروتئین هایی که با پروتئین و دی ان ای و آر ان ای برهم کنش میدن به وفور به نسبت موجودات دیگه بالا رفته و به یک نوعی پویایی ژنوم در انسان بالاتر از موجودات دیگه است. اگر توی این تصویر دوباره مقایسه بکنیم ژنوم انسان رو با 4 تا موجود مدل دیگه از جمله همین نماتود استنورپتیتیس، ما می‌بینیم که اون تو قسمت فانکشن یا فعالیت‌ها ژن‌هایی که فعالیت‌هاشون برهم کنش دارن با مولکول دی ان ای که نوشته دی ان ای بایندینگ، توی انسان بالای 2000 تا ژن هست که چنین پروتئین‌هایی رمزدهی می‌کنن، ولی توی این نماتودی که نام بردیم حدود 295 تا، یعنی تقریباً حدود 1/7 نسبت به انسان یا 1/8 نسبت به انسان کمتر هست این نوع ژن‌ها توی این موج و توی موجودات دیگه هم مقایسه می‌کنیم باز اینها به نسبت توی انسان خیلی بالاتر هست به نسبت گیاهان، مخمر و مگس سرکه و ژن‌های هوموباکس توی تکوین و تمایز ژنی نقش دارن. باز هم می‌بینیم که توی انسان به نسبت این ژن‌ها تعدادشون بالاتر هست و ژن‌هایی که در کارکرد سلول‌های عصبی نقش دارن که ما می‌بینیم توی انسان، حالا من با کادر سبز نشون دادم 70 تا 15 تا و توی موجودات دیگه به ندرت ما می‌بینیم که این ژن‌ها وجود داشته باشن. بنابراین یکی از در واقع عوامل تمایز انسان کارکرد ژن‌ها هست، یعنی پروتئین‌هایی کد میشه که کارکردهای متفاوتی دارن. بنابراین نکته اول اینجا هستش که بحث بیان ژن مطرح هست و اینکه این پروتئین‌هایی هستن که برهم کنش و پویایی عوامل داخل ژنوم رو بالا بردن و دیگر اینکه اون ارتباطات بین سلولی به خصوص در سیستم عصبی توی انسان تقویت شده. توی این تصویر اگر نگاه بکنیم خب توی ژنوم انسان چی هست؟ توی ژنوم انسان حدود 1.5% توالیه‌هایی هستن که این توالیه‌ها کددهی می‌کنن به آمینو اسید و در نهایت به پروتئین. بیش از 98.5% ژنوم انسان در واقع عواملی هستن که رمزدهی به پروتئین نمی‌کنن، غیر رمزگذارند یا نان کدینگن که حالا مقایسه بکنیم اینها رو توی ژنوم حدود 63% شون توالیه‌های تکرار شونده‌ای هستن که توی سرتاسر ژنوم وجود دارن و برای هیچ پروتئینی رمزدهی نمی‌کنن و ما اینها رو قاعدتا طبق تعریف ژن نمی‌دونیم، ولی بهش میگیم نواحی غیر رمزگذار و توی ژنوم بیشتر ژنوم رو توی انسان به خودشون اختصاص دادن. در 2 دهه گذشته دانشمندان اومدن گفتن که این ماده تاریک ژنومه، چطور ژنوم 1.5% به پروتئین رمزدهی میکنه پس مابقیش داره چیکار میکنه؟ ما اینجا اگر نگاهی به یکی از بخشی از کروموزوم یک انسان بکنیم، نواحی که ژنی هستن رو توی این تصویر ما به رنگ سبز می‌بینیم روی این ژن‌هایی که 2 تا ژن اینجا اسامیشون هست BSND، PKSK9، USP24. نواحی که سبز هستن نواحی اگزونی، یعنی جاهایی هستن که به آمینو اسید رمزدهی می‌کنن. مابقی نواحی بین ژنی هستن و توالیه‌های تکرار شونده که با رنگ‌های غیر سبز ما می‌بینیم و توی ژنوم انسان به نظر میرسه که بیشتر ژنوم انسان تحت سیطره این توالیه‌ها هست، نه سیطره توالیه‌هایی که رمزدهی می‌کنن به آمینو اسید. اینجا اگر مقایسه بکنیم این بخشی از ژنوم انسان رو با 3 تا جاندار دیگه که مخمر نان هست، مگس سرکه هست و گیاه ذرت، ما می‌بینیم که اینجا هم باز همون رنگ سبز نشان دهنده تراکم ژنی هست و توی انسان این تراکم پایینه با نواحی بین ژنی پر شده بیشتر ژنوم انسان. توی مخمره که موجود تک سلولی هست به شدت نواحی بین ژنی کمه و ژن‌ها تراکم دارن و توی حشره مثلا مگس سرکه باز می‌بینیم نواحی بین ژنی زیادن و توی گیاه هم همینطوره منتها نوع توالیه‌های بین ژنیشون و توالیه‌های تکرار شونده‌شون با هم متفاوت هست. پس یک اهمیتی باید داشته باشن این نواحی بین ژنی. و اگر نگاه بکنیم به تراکمشون ژن دنسیتی رو مثلا توی این جدول نگاه بکنیم می‌بینیم توی مخمر به ازای هر مگابیت 549 ژن وجود داره در صورتی که برای انسان 6 تاست، یعنی تراکم ژنی توی انسان پایین اومده و تعداد اینترون ها که در واقع ژن ها رو گسسته میکنن توی انسان 8 تا، توی مخمر 500، یعنی توی انسان این توالی ها به شدت بالا رفتن و نواحی بین‌ژنی هم که داریم میبینیم توی انسان نواحی بین‌ژنی 44% ژنوم انسان رو به خودشون اختصاص دادن. خب این نواحی بین‌ژنی اهمیتشون در چی هست؟ اهمیت اینها در بیان ژن و در بکارگیری اون ژن‌هایی هست که رمزدهی میکنن به پروتئین یا ژن‌هایی که رمزدهی میکنن به انواع آرناهای فانکشنال. این آرناهای فانکشنال چی هستن؟ ببینید توی ژنوم بخشی از ژنوم که رمزدهی میکنه به پروتئین کد میده برای mRNA، ولی مابقی ژنوم انواع آرناهایی میکنه که تولید میکنه که بهشون نان کدینگ RNA میگیم. توی این تصویر بیش از 96% ژنوم رمزدهی میکنه به آرناهای نان کدینگ که حالا بخشی از اینها آرناهای ریبوزومی هستن، آرناهای ترانسفر هستن و شورت نان کدینگ و لانگ نان کدینگ آرناهایی که برای دانشمندان پرسش رو مطرح کرد که این همه RNA که رمزدهی به پروتئین نمیکنه اینها چیکار میکنن. و توی این تصویر ما میبینیم که تعداد این آرناها که حالا نان کدینگ آرناها، ام آی آرناها، آر آرناها، اس ان آرناها، استون آرناها و دیگر نان کدینگ آرناها توی ژنوم بسیار بالا هست و حتی تعدادشون از تعداد ژن ها بالاتره. بنابراین ما تعداد آرناهایی که توی ژنوممون بیان میشه بسیار بیشتر از تعداد ژن هایی هست که توی ژنوممون بیان میشه. و اینها از کجا بیان میشه؟ جانم
زمانتون تموم شده.
من سعی میکنم سریعتر پیش برم، سعی میکنم زودتر تمومش کنم. این
مرسی.
آرناها از هر جای ژنوم میتونن بیان بشن و فانکشن های متفاوتی توی سلول دارن، عملکردهای بسیار گوناگونی دارن و اینها به چه عنوان عمل میکنن؟ به عنوان بخشی از اپیژنوم یا اپیژنتیکی که تسلط داره بر ژنوم. و اپیژنوم اجزای متفاوتی داره. اپیژنتیک مواردی که دی ان ای رو تغییر میدن، اصلاح میکنن، یا روی کروماتین تغییراتی ایجاد میکنن، یا نان کدینگ آرناها رو پدید میارن، یا در مودیفیکیشن آرناها نقش دارن و اینها پویایی و برهمکنششون بسیار زیاد هست. به همین خاطر به نظر میرسه مکانیزمی توی ژنوم وجود داره تحت عنوان مکانیزم بکارگیری تنظیم شده و کنترل ترکیبی که اینها برای تکامل موجودات پیچیده‌تر پدید اومدن و این نشون میده که برای تکامل یک موجودی مثل انسان نیاز به ابداع انواع متفاوتی پروتئین نبوده، بلکه این سازوکار بکارگیری ژنوم و سازوکار پویایی ژنوم هست که تکامل پیدا کرده و میتونه این ژنوم رو به شیوه‌های متفاوتی بیان بکنه و ازش هر لحظه چیزهای متفاوتی بیرون بکشه. بنابراین یک نوع اختصاصیت توی زیست‌شناسی وجود داره که این در برهم‌کنش با محیطه و این ساختار تنظیمگر ژنوم نه تنها توی خود ژنوم وجود داره، توی سلول هست و توی محیط هم هست که این مجموعاً توالیه‌های رمزدهنده، ماشین تنظیم کنندگی و اون زیست‌بومی که فرد توش داره زندگی میکنه تاثیر داره بر بیان ژن‌های داخل این ژنوم. بنابراین ژنوم صرفاً مجموعه‌ای از ژن‌ها نیست، بلکه مجموعه‌ای از ژن‌ها هست و انواع نانکدینگ دی ان هایی که فانکشن‌های بسیار متفاوتی دارن و توی تکامل نقش بسیار بالایی بازی کردن. و در نهایت جمع‌بندی این هستش که اون چیزی که کلیدی هست توی تکامل انسان تعداد ژن‌ها یا اندازه ژنومش نیست، بلکه نحوه بیان ژن‌ها هست و چطور این ژنوم به کار گرفته بشه که این برمیگرده به نواحی بین‌ژنی و تکامل اپیژنتیکی که رمز تکامل در انسان هست. این دیدگاهی هست که تا به امروز حالا میشه گفت به یک جمع‌بندی دانشمندان بهش رسیدن که نیاز به کارهای بسیار بیشتری داره که ما درک کاملتری از این داشته باشیم. من از همه شما سپاسگزارم و اگر اشکالاتی توی ارائه بود از همه حضار عذرخواهی میکنم.
خیلی ممنون دکتر سلطانی، متشکرم ازتون. مارک، can you hear me?
Yes, I can hear you.
در خدمت پروفسور مارک استون کینگ هستیم، استاد و رئیس گروه انسانشناسی تکاملی در پژوهشکده ماکس پلانک و از مهمترین نظریه پردازان در مبحث تکامل و پراکنش انسان هستند که خیلی لطف کردن و دعوت ما رو پذیرفتند. So we are ready, we can start.
All right, so thank you for that introduction, and I would like to thank the organizers for the invitation to participate in this event. I'm always happy to do whatever I can to help out with promoting evolutionary biology, especially human evolution, anywhere around the world. So I'm going to start with some history, which may very well be an indication of how old I'm getting, but I do happen to think that history is important, that if we want to know where we're going, it helps to know where we've been. And so I'm going to start with this question that kept a lot of us busy for a good many years, and that is the question of how did our own species, anatomically modern humans, originate? And I want to point out that it's a bit deceptive because it makes it sound like we're interested in a single entity, a single thing, anatomically modern humans. But actually what we want to know is when we look around the world, we see what looks to us like an incredible amount of diversity in people's appearances and their skin color, hair morphology, height, weight, as well as their culture, right? Languages and other cultural traits. So we'd like to know, how did all of this diversity originate? Is it recent? Is it ancient? Is it single origin? Or is it multiple origin? Back when I was a graduate student in the 1980s, there were basically three different models. I mean, there have always been lots of different models proposed for human origins, but there are mainly three that most people were discussing and debating. And so one of these shown here is multi-regional evolution, proposed originally based on the fossil record. And according to the fossil record, everything that happened in our evolutionary past was in Africa up until about 2 million years ago. And then beginning around 2 million years ago or so, the first hominins began spreading out of Africa, and they spread across the entire old world, and they're quite successful. But what spread out at 2 million years ago wasn't anatomically modern humans, it was something else. Here it's called Homo erectus. Different anthropologists would give different names. The names don't really matter. What's important, though, is that then it's only around 350,000 years ago that anatomically modern humans appear on the scene. And so while according to multi-regional evolution, the main lines of evolution are descent within each of the major regions of the old world. So modern Africans are mostly descended from ancient Africans, modern Europeans mostly from ancient Europeans and so forth. You also have these, so that's indicated by the vertical arrows, but you also have these horizontal arrows, which indicate gene flow, which was very important then for keeping the entire old world population of hominins spread from sub-Saharan Africa to Western Europe to Eastern Asia, even out into Oceania, evolving in concert to become anatomically modern humans. Now, as opposed to multi-regional evolution, there were a variety of theories that proposed a single origin, a single place, a single time for modern humans. The most prominent of these was Africa. So again, according to the fossil evidence, you have the first hominins spreading out of Africa 2 million years ago. They're successful for hundreds of thousands of years. But then somewhere around 300 to 350,000 years ago, something special happens in Africa, and that something special is us. And then beginning somewhere between 50 and 80,000 years ago or so, our ancestors spread across Africa and they began spreading out of Africa. And in the most extreme version of the out of Africa hypothesis, which is out of Africa with complete replacement, these archaic non-African hominins are replaced completely. They don't contribute any ancestry at all to modern humans. So modern humans today are basically all African refugees, if you will. And then the third hypothesis is out of Africa, but allowing for some contribution of ancestry to modern humans from archaic humans outside of Africa. And there was debate about where this might have occurred. Was it in Europe with Neanderthals, or was it elsewhere with other archaic hominins? But the crucial difference is that it was not a complete replacement, but rather that there was some ancestry contributed by the non-African hominins. Now, these were all proposed originally based on fossil evidence, but they proved incredibly difficult to differentiate between these different hypotheses based solely on the fossil evidence. What it took was genetic evidence, because if you think about it, what these different hypotheses do is they make different predictions about what we should see in terms of African ancestry in non-African populations. We can put this on a continuum. So down here we have multi-regional evolution, which would say that some of the important mutations involved in becoming humans occurred in Africa, but others occurred elsewhere. And anyway, regardless of where they occurred, they would be spread around by gene flow so quickly that we wouldn't expect to see any signal of where they originally occurred. And then at the one extreme, we would have African origin with complete replacement, which would say that we're basically all 100% Africans in our ancestry. Then in between would be the assimilation hypotheses, which would say that most of our ancestry is African, but some small, maybe 1, 2, 5%, but some small but detectable amount of our ancestry should be have been contributed by these some non-African archaic hominin group. So this says if we want to distinguish among these hypotheses, we should look at the genes. And the first gene to be looked at any sufficient detail to try to address these different hypotheses was this one. It's mitochondrial DNA. It's found outside the nucleus of the cells in the mitochondria. It has a number of unusual and useful properties, one of which is that it's transmitted from mothers to sons and daughters. Males have mitochondrial DNA, but they don't transmit it to their offspring. So by studying variation in mitochondrial DNA, we're learning about the maternal history of human populations. And the first in-depth study of human mitochondrial DNA variation was carried out by Rebecca Kahn and myself when we were graduate students in Alan Wilson's lab. And we produced this sort of now iconic phylogenetic tree of mitochondrial DNA lineages in different human populations, where the first split in the tree separates a branch only of Africans from everyone else in the world, including some African individuals. And the simplest explanation for this pattern would be that the ancestor was African, left some descendants in Africa, and other descendants went out of Africa. There's also a scale here, the amount of sequence divergence. So this is basically an estimate of how much mutational diversity has arisen since the common mitochondrial DNA ancestor. And using a molecular clock approach, we can estimate that this amount of mutational diversity corresponds to around 140 to 280,000 years. So mitochondrial DNA indicated a recent African origin. Now, this study was met with a lot of controversy, with a lot of debate, a lot of criticism, some of it justified, some of it not. But further studies have basically upheld that same picture. So the picture we have today of mitochondrial DNA, now using complete mitochondrial DNA genome sequences rooted with the chimp, is that we get a phylogenetic tree very similar to what we saw before, and that it would take about 150,000 years to produce all of the mitochondrial DNA diversity that we see in modern humans today. So mitochondrial DNA clearly indicates a recent African origin. But mitochondrial DNA is just one gene, and we know that the single gene history can differ from that of the species because of chance events, or perhaps because there's been selection. So we need to look at other genes. The next gene to be looked at in sufficient detail to address these questions was this tiny, pathetic little chromosome here. This is the human Y chromosome compared to an X chromosome. It's what makes us males male. And it's the sort of paternal analog to mitochondrial DNA in that it's transmitted from fathers to sons. So it provides insights into the paternal history of populations. And studies of Y chromosome variation in modern humans produced trees that look very much like the mitochondrial DNA tree, with primary division separating Africans from other groups, and that it was taken around 200 to 250,000 years to produce all of the Y chromosome variation that we see in modern humans today. So the Y chromosome also has a strong signal of a recent African origin. And of course, nowadays, we're well beyond these single gene sort of studies. We can look at genome-wide variation. We can look at variation across all of the chromosomes that we have. And so when we do that, we invariably get trees that look much like the mitochondrial DNA and Y chromosome trees, where the first splits involve African populations. Moreover, with genome-wide data, we can do a lot more than we can do with just mitochondrial DNA or Y chromosome data. So one thing we can do, for example, is we can use computers to carry out simulations of different population histories, different demographies, and ask what simulated population history gives the closest fit to the real genome-wide data that we observe today. And those simulated histories always, without exception, involve a recent African origin of modern humans. So it's not just mitochondrial DNA, it's not just the Y chromosome, it is genome-wide that there is a strong signal of a recent African origin. And so studies of genetic variation in modern human populations, I think, and I think most people would agree, pretty convincingly rule out multi-regional evolution. But what about out of Africa with complete replacement versus out of Africa with assimilation? And here, the studies based just on modern human populations were really not able to distinguish between these two hypotheses. You would occasionally get a study which said, oh, here's a very strange pattern of variation. This looks like this must have been something that came from Neanderthals. And then that would be followed up by a few months later by another study saying, well, actually, you can get the same pattern with out of Africa and complete replacement. So the argument went back and forth and back and forth. But we do now have an answer as to which of these is the best fit best model for human origins. And that comes from additional data, not from modern humans, but genome sequences directly from archaic humans, so Neanderthal genomics. So the first Neanderthal genome sequence was produced in 2010. It was a very messy sequence. It was very low coverage, lots of errors, but it was sufficient to address this question of whether or not there's any Neanderthal ancestry in modern human populations. So it was a composite of DNA sequences from 3 different bones, all coming from this cave here, Vindija Cave in Croatia, and they're all dated to in excess of 38,000 years ago. Now, there were 3 different analyses done to address this question of whether or not there's Neanderthal ancestry in modern humans. All 3 gave the same result, so I'm just going to walk you through one of them, and I'll skip the gory mathematical details, but I want to give you some idea of the logic that's involved here. So one prediction we can make is that if there was admixture, then the Neanderthal sequence should match alleles in some humans that descended from the admixture event significantly more often than in others who are not descended from the admixture event. So the test is very simple. We compare 2 human sequences with different alleles, one of which is the ancestral state, one of the other which is the derived state. And we can tell which is ancestral and which is derived by looking at sequences in non-human primates. And that tells us which is the ancestral allele when we have a polymorphism, when we have a difference in the sequence, for which the Neanderthal has the derived allele, okay? Now, if we take 3 different sequences and compare a Neanderthal to 2 humans, just 2 humans at random, say Donald and George, most of the time what we will find is that we will see that the Neanderthal has the allele and both humans have the derived allele, and that would reflect a mutation that occurred after the separation of Neanderthal lineage and before the divergence of the human lineages. Or we might see that the Neanderthal has the derived allele and the 2 humans have the ancestral allele, and that would reflect the mutation that occurred specifically on the Neanderthal lineage. And when we take a Neanderthal sequence and 2 human sequences, and we just look for positions that are different, vast majority of the time, this is what we see. But occasionally we see discrepancies between the gene tree and the species tree. So these are cases where the Neanderthal has a derived allele and one human has a derived allele, but the other human has the ancestral allele. There's 2 possible explanations for this mismatch. One is ancestral polymorphism, that the mutation from the ancestral to the derived allele occurred in the ancestral population of Neanderthals and modern humans. And then by chance, the Neanderthal gets a derived allele, one human gets a derived allele, the other human gets the ancestral allele. The other potential explanation is admixture with Neanderthals. And so this would be a mutation from the ancestral to the derived allele that occurred on the Neanderthal branch. And then the Neanderthals contributed this derived allele to some humans, but not to others by admixture. How can we tell the difference between these 2 potential explanations? It's pretty straightforward. In the case of ancestral polymorphism, we expect the Neanderthal to match the 2 humans with equal frequency. That is, we expect Donald and the Neanderthal to have the derived allele just as often as the Neanderthal and George have the derived allele. It's basically like a coin flip, which one gets the derived allele, which one gets the ancestral allele. In this case, we would expect Donald to match more significantly often to the Neanderthal than George would. So that's the basic of the test. We compare diverse human genome sequences from around the world and ask if one shares more alleles with the Neanderthal. If we can take 2 Africans, we see no excess matching with the Neanderthal. If we take 2 non-Africans, no matter where from around the world, we also see no excess matching with the Neanderthal. But where things get interesting is when you compare an African to a non-African, then you always see, without exception, an excess of matching of the non-African to the Neanderthal. And what's interesting is it doesn't matter where the non-African is from. It could be a French individual as shown here, which would make sense because Neanderthals were in Europe, along with the ancestors of French people. But you could take a Han Chinese, you could take a Papua New Guinean, you could take someone from the New World. They all show an excess of matching compared to an African to the Neanderthal. And so that's sort of summarized here, that we see this excess matching. Now, it turns out there's 2 possible explanations, though, for this excess matching. One would be that when the ancestors of modern humans left Africa, they fairly quickly encountered Neanderthals, interbred with them, picked up Neanderthal ancestry, and then started moving out elsewhere into the old world and diverging from one another. And so there would be this shared signal of Neanderthal ancestry in all non-African populations. But the other possible explanation is that there was ancient structure within Africa. Now, this is a little hard to explain, but the basic idea is that Neanderthals also came out of Africa several 100,000 years before modern human ancestors did. So if the African population that gave rise to the ancestors of Neanderthals also later gave rise to the ancestors of modern humans, then we would expect modern humans outside of Africa and Neanderthals to also share some excess alleles, not because of interbreeding, but just because of the shared ancestry, common ancestry. The test I just told you about doesn't distinguish between these two, doesn't tell the difference, but there are other tests one can do that do. And when applying those other tests, this explanation is pretty convincingly ruled out. And so we're left with the explanation that there was indeed interbreeding between Neanderthals and the ancestors of all non-Africans. So with the aid of Neanderthal genomics, we can rule out of Africa with complete replacement, and we're left with out of Africa with assimilation, where there's around 1 to 2% Neanderthal ancestry in non-Africans. Now, in the course of doing this work, Svante Pääbo's group, who was leading the project, the effort, were trying to get their hands on any fossil that might potentially be a Neanderthal to see if it would have enough DNA to produce a genome sequence. And one of the fossils they screened was this tiny little fingertip bone that came from this cave. This is Denisova Cave, located here in the Altai region of southern Russia. So you can see how small it is. And when screening the mitochondrial DNA, Neanderthals have different mitochondrial DNA than modern humans. And so it's a good screen to tell whether you're dealing with a human or a Neanderthal fossil. They instead got an unexpected result because it was not a modern human sequence, but it was also not a Neanderthal sequence. It was something that had never been seen before. So here's a tree of modern human mitochondrial DNA sequences. Here in blue are some Neanderthal mitochondrial DNA sequences, again, falling well outside the range of modern human diversity, diverging maybe 500,000 years ago from the modern human mitochondrial DNA sequence. Here's the Denisova, it's twice as divergent about 1 million years ago. But remember what I said about the history of a single gene not necessarily reflecting the history of a species, and this is an example, because when you do the genome sequence from that Denisovan fingertip bone, you find out that Denisovans are actually a sister group to Neanderthals. And the divergence between the Denisovans and the Neanderthals is slightly bigger than the biggest difference that we see in genome sequences from modern humans today. So Denisovans are then this sort of novel hominin group that we knew nothing about before we had this genome sequence. It's a sister group to Neanderthals. Now we can ask, did they interbreed with modern humans? And so we do this matching statistic I told you about. Compared to Eurasians, no excess matching, a Eurasian to an African. There is excess matching of the Eurasian to the African, but it's about what you would expect given that the Denisovans are a sister group to Neanderthals. So nothing peculiar there. Where things get interesting, though, is when you compare a Melanesian, someone from New Guinea or Australia, to a Eurasian, whereas with the Neanderthal, you don't see excess matching. With the Denisovan, you see excess matching of the Melanesian, and that also holds in Africa as well. And so what it looks like there's been, in terms of a very simple model, is that there's been 2 episodes of interbreeding between archaic and modern humans. One from Neanderthals into the ancestor of all non-Africans, the other from Denisovans specifically into the ancestor of Melanesians. But if there's one thing we've learned about human evolution, is that simple models are not likely to be correct. And what we know now from a decade of further research is that there have been multiple episodes of interbreeding between modern humans with Neanderthals. So here in this tree, you see the Neanderthal gene flow into the ancestor of all non-Africans. And you also see the Denisovan gene flow into the ancestor of Melanesians. But it turns out that Europeans and East Asians have a little bit more Neanderthal ancestry than Melanesians do, suggesting additional gene flow there. And the human populations that have the highest amount of Neanderthal ancestry are not Europeans, as you might expect, but rather East Asians. And so it looks like they may have also experienced a little bit of additional Neanderthal gene flow. We're also finding multiple episodes of interbreeding with Denisovans. East Asians originally thought not to have Denisovan ancestry. Now we know they have a very tiny amount of Denisovan ancestry, around 0.2%. And moreover, that seems to reflect at least 2 different pulses of Denisovan gene flow in East Asians. It's really remarkable. We also see this in Papuans now, at least 2 pulses of Denisovan gene flow in Papuans. And the most recent of these is dated to about 30,000 years ago. We're in New Guinea. And so it suggests, it's quite controversial, but it suggests Denisovans may have been in New Guinea, been able to make the water crossing to get into Australia and New Guinea. And we also see Denisovan ancestry in the Philippines. And some of these Philippine groups have the highest amount of Denisovan ancestry recorded. And it's clearly different ancestry than what we see in the Philippines. So many different pulses of Denisovan ancestry in modern humans. We're also finding other Neanderthal ancestry. So this is a fossil called Awasi 1. It's dated to about 40,000 years old, comes from this cave in Romania. It's been described as being a modern human, potentially with some archaic features. Turns out to have a whopping 6 to 9% Neanderthal ancestry compared to 1 to 2% that you would find in you or me today. And so this means that the Neanderthal interbreeding in the ancestry of this individual occurred about 4 to 6 generations prior to this individual being born. So very close in time to the interbreeding event. And even more interesting, if you look at that Neanderthal ancestry, it's not the same as the Neanderthal ancestry that we see in modern humans today. So it's an example of an interbreeding event between modern humans and Neanderthals that didn't go anywhere, that went extinct. Probably there was a lot of that went on. We're also starting to detect modern human ancestry in Neanderthals. The first high coverage, high quality Neanderthal genome sequence came from the Altai region, actually came from Denisova Cave, where there were both Neanderthals and Denisovans. This Altai genome sequence has about 1% modern human ancestry. Now, nobody thought much about that because you always have a small amount of modern human contamination in these Neanderthal fossils from the people who handle the fossils or maybe from the laboratory manipulation. But then Martin Colville, a postdoc in Svante's group, got the bright idea to, well, let's take a look at that modern human ancestry and just see what does it look like. Lo and behold, it turns out it was not related to modern humans, as you might think. Instead, it branches off prior to all of the divergence of modern human genome sequences. So it reflects a modern human population that existed prior to the out of Africa migration from which all modern populations are derived out of Africa today. So it suggests that there was an earlier migration of modern humans from Africa that contributed ancestry to Neanderthals, but did not contribute any ancestry to modern populations. And we're even detecting admixture between Denisovans and Neanderthals. Now, whenever paleontologists excavate a cave, they will end up with hundreds and thousands of very tiny little bone fragments that are not diagnostic. You have no idea what sort of creature they might have come from. And usually they're just put in a bag and put aside. Now, in principle, one could take those fragments and do ancient DNA analysis, but given the cost and the amount of time it takes to do this, it would be prohibitive to try to screen hundreds to thousands of samples. You just can't do it. But a few years ago, the method was developed to look at not at DNA, but at ancient proteins. And with ancient proteins, you can tell what family you're dealing with of mammals. And so in one of these studies, 2,400 bone fragments from Denisova Cave were screened by this method, this mass spectrometry method called zooms. One of those was identified as being a hominid. So now you do the ancient DNA on that one fragment, and here it is in all its glory. And remarkably, the genome sequence indicates that this individual had a Neanderthal mother and a Denisovan father. And there's also some indication that the Denisovan father had some Neanderthal ancestry as well. So I think if you want to sum up what we've learned about human evolution from archaic genomes, it's this. Everyone was having sex with everyone else. And that's what characterizes human evolution. Now, this archaic ancestry also is a new source of information, not only on the mating habits of our ancestors, but on selection and adaptation that occurred during human evolution. So here is a cartoon of the 22 human chromosomes with the red and blue bars indicating Neanderthal ancestry found in Asians and Europeans. And what you can see is that Neanderthal ancestry is not distributed at random across our chromosomes. Instead, it's clumped. And there are some regions that have significantly less Neanderthal ancestry than expected. These are called deserts. And so presumably, these reflect regions of the genome where Neanderthal variants don't work well with modern humans, that there's something significantly different between modern human and Neanderthal genome sequences in these regions, something of functional interest. And so if you start to look at the genes that occur in these deserts, one of them is a gene called RNX2, codes a transcription factor that is involved in turning on expression of many other genes. And mutations that eliminate the function of RUNX2 result in a disease syndrome in modern humans called cleidocranial dysplasia, or CCD. And some of the features of this syndrome are you get delayed closure of the cranial sutures of the bones of the skull. You get frontal bossing, this protuberance around the eyebrows. You get abnormal shoulder bones and collar bones. You get a bell-shaped rib cage, and you get dental abnormalities. And what's interesting about these features is they recapitulate some of the skeletal differences between modern humans and Neanderthals. So Neanderthals, for example, have this protuberance over the eyebrow ridge called frontal bossing. Modern humans don't have it. Individuals with CCD have this. Modern humans, we have a barrel-shaped rib cage. So our rib cage is vertical, going from top to bottom. Neanderthals have a bell-shaped rib cage. It flares out going from top to bottom. Individuals with CCD have a bell-shaped rib cage. They also have abnormal collarbones and abnormalities in the shoulder bones. And these are exactly the sorts of things that you see in Neanderthals. So we've learned something then about at least several of the genes that are involved in the differences between Neanderthals and modern humans by looking in these deserts. But now, in addition to desert, there are also islands of Neanderthal ancestry. These are regions of the genome with significantly more Neanderthal ancestry than expected. And so if we look at what sorts of genes occur in these islands, we find that they occur in a couple of different functional categories. So they include genes that are involved in the immune response and how we respond to parasites and to disease-causing organisms, bacteria and viruses. They are involved in skin morphology, skin pigmentation, and they are involved in a diet, how we respond to different dietary sources. And if you think about it, I think these all sort of make sense, right? These are ways in which we have to interact with our environment. In Neanderthals, they came out of Africa several 100,000 years ago. Now they're in this new environment, new climate, new disease organisms, new sources of nutrition. They have to adapt to all these novelties, and they do so because they're successful for hundreds of thousands of years. And then our ancestors come out of Africa, and they have to do the same thing all over again. So maybe by this adaptive introgression, we picked up selective variants that were already proven to be good in this novel environment. So who knows, maybe without this adaptive introgression, maybe our ancestors would not have been able to survive outside of Africa. It's interesting to think about. So these all sort of make sense, that these would help modern humans adapt to the new conditions outside Africa, that these would be selected for. But there's some other signals that are a little harder to explain. So one of these is in a gene called EPAS1. It turns out this is a gene that's been well known to be involved in high altitude adaptation in Tibetans who live in altitudes in excess of 4,000 meters. So these colors show you the different haplotypes, the different variants found at this gene in different human populations. The TIB population is Tibetans, and you can see they have a very high frequency of this variant, which is the selected variant that works well at high altitude. You ask, where else do you find this? Hardly anywhere else in modern human populations. But it turns out that this matches the Denisovans. And in fact, it looks like Tibetans got this adaptive variant from Denisovans. Now, that seemed really hard to believe that Denisovans could be living at such high altitude. But further support for this came a few years ago from a study. It was actually not a new fossil find. It's an old fossil find that's been newly analyzed. It's a jawbone, as you can see, that came from the Spashia Karst cave, been dated to about 160,000 years old. Tried to get ancient DNA from it, but this is too old. There's no surviving authentic DNA there. But look at the proteins. The protein suggests it is indeed Denisovan. And in a follow-up study that looked at DNA from the cave sediments, there's Denisovan mitochondrial DNA in the cave sediments. So yes, it looks like Denisovans were indeed capable of living at such high altitudes, which then, makes you sort of wonder, so what were these Denisovans anyway? Could they have been the source of the myths about the abominable snowmen? So I'd like to conclude now with take-home messages. So in addition to telling us about the sexual habits of our ancestors, the archaic genomes are a source of great new insights into the dispersals of modern humans. They tell us about the genetic differences that make us human, and they tell us about alleles of functional importance that we received from archaic humans. So I'll end with the acknowledgements. The work that I talked about on Neanderthal and Denisovan genomes was the work of a great many people from a great many institutes. These are the main players at the Max Planck Institute for Evolutionary Anthropology in Leipzig, and their fearless leader, Svante Pääbo. As you probably know, Svante was awarded the Nobel Prize last year for his discoveries concerning the genomes of extinct hominins and human evolution. And I think it's just a great, great acknowledgement of the importance of ancient DNA and human evolutionary studies for not just medicine and physiology, but for evolutionary genomics and for humans in general. And of course, we also have to acknowledge the Max Planck Society for the funding that makes all this work possible. And finally, if you'd like to know more about molecular anthropology and what we've learned using molecular approaches to address anthropological questions, there is a book I can recommend. And with that, I'll end, and thank you.
And thank you again for accepting our invitation. Hello, Bernard. Can you hear me? خب در خدمت پروفسور برنارد وود هستیم، استاد تکامل انسان و آناتومی تکاملی انسان در دانشکده انسانشناسی دانشگاه جورج واشنگتن که خیلی لطف کردن و دعوت ما رو پذیرفتن و سخنرانیشون رو ما الان میشنویم. We are ready. Take the ownership of your slides and go on. Thank you.
Thank you very much for the invitation. And thank you also to Mark for just a splendid talk. My talk is going to be a little, it's going to try to answer two questions. And obviously these questions relate to Charles Darwin, and I will explain exactly how they relate to him in a minute. The first of the questions is, who are our closest living relatives, and when did we share a recent common ancestor with them? The second question relates to the past and is a slightly longer question. Does our twig of the tree of life consist of just the ancestors of modern humans, or do we also have close fossil relatives that did not make it onto the surface of the tree? In other words, they are extinct. The questions come from the two major ideas in the origin of species. One of the two major ideas in the origin of species. The two major ideas are natural selection, and the other one is the notion of common descent. The tree of life is an image that I'm sure the people attending today are familiar with in ancient literature. But what Darwin was suggesting was the tree of life was no longer just a metaphor, a visual metaphor, it was actual. So the first question is, who are our closest living relatives, and when do we share a recent common ancestor with? And there are various, and so on the left is a modern human. Next to them are the two chimpanzees, the common chimpanzee and the bonobo, and then the mountain gorillas and lowland gorillas. And the two main species of orang, one from Sumatra and one from Borneo. Now, when I was a student, we were told that there was such a major difference between modern humans and all those apes that you put the humans in one family, the Hominidae, and you put all the apes in another family called the Pongidae. But what is the situation as opposed to what was I taught as a student? And there are various lines of evidence that you can use. One of them is what I'm calling traditional morphology. In other words, what do the bones look like? What do the soft tissues look like? And the first person to investigate this was a man called Edward Tyson, who was a doctor. He was medically qualified. He was a member of the Royal College of Physicians. And he was actually the medical superintendent of the Bethlehem Institute for the Insane. And Londoners used to shorten the Bethlehem Institute for the Insane to Bedlam. And that is a word that we use when we think there's much too much noise. And so you accuse your children or the students of creating Bedlam. Tyson was a morphologist who was the person in the Royal Society who would be sent organisms that were exotic and should be dissected. And one of the organisms that he was sent in the late 1600s was a creature, and the skeleton of which is on the image there. He didn't know what it was, but it was an infant chimpanzee that had been brought back from Africa by some merchants to Bristol. And he dissected it, and the report of his dissection is a magnificent report. He dissected it along with another anatomist called Cowper. And their dissection was meticulous. And they found that there were very few differences between this chimpanzee and the anatomy of modern humans. The next recorded dissections of a chimpanzee after Tyson were by a man called Traill and by Richard Owen. But it was Thomas Henry Huxley who moved the debate forward when he did some dissections of chimpanzees. And what Huxley did was that he provided additional morphological evidence to that already accumulated by Tyson and Traill and Owen, and in Evidences to Man's Place in Nature in 1863. He argued for a close relationship between modern humans and the apes, especially the African apes. And what he wrote in Evidences to Man's Place in Nature is whatever part of the animal fabric might be selected for comparison. The lower apes, the monkeys, and the gorilla, and there are various reasons why he focused on the gorilla, would differ more than the gorilla and the man. In other words, what he was saying was that the apes and man, human beings, were more similar than the apes and monkeys. So what is the situation today with regards to traditional morphology? Not long ago, I had a graduate student called Rui Diogo, and what he did for his PhD thesis was to look at the morphology of the striated muscles. These are the muscles that move your limbs. These aren't the muscles in your gut or your heart muscle. And when we looked at the striated muscles, and we looked at what they look like in modern humans and in chimpanzees, and latterly in bonobos and in gorillas and orangs, it was obvious that the muscles, the evidence from the muscles, linked the African apes together, gorilla, modern humans, and chimpanzees. So you can see here that the striated muscles in the gibbons, which is the top row, are very distinctive, which is probably not surprising given their very unusual locomotion. And then there are shared features in the common ancestor of orangs and the African apes and modern humans. There are more shared features in in the common ancestor of the African apes and modern humans. And there are a few shared features in modern humans and chimpanzees. So striated muscles reinforce the results from Tyson and the people who came after him. So what about the molecular evidence? Well, the molecular evidence was investigated originally by two researchers. One was called Emil Zuckerkandl, and what he did was that he broke a rather complicated protein called hemoglobin down into its component peptides, and then he used a relatively modern method called electrophoresis, or modern at the time, to see if he could separate the peptides. And when you visualize the peptides, when you spray material onto the gel, you can see that the patterns of modern humans and gorillas are almost indistinguishable. But they can be distinguished from the gorilla and the orang, and especially the rhesus monkey. The other line of evidence was pursued by Maurice Goodman, who used what was then the relatively new method of immunology to explore the difference between the albumins of modern humans and the African apes and the orangs and monkeys. And basically, what you have to notice in this, there are wells. There are two wells in the top of each of these, and there is a single well in the bottom. And you put antibodies in the bottom well, and you put the albumins in the top well, and they move through the gel towards each other. And the more of a spur there is in the white line, which is where the interaction occurs, the more of a spur there is, the greater the difference. And you can see on the top right that when you do that between a chimpanzee and a man, and you offer those two, you can see that there is no spur. In other words, this particular assay cannot discriminate between modern humans and chimpanzees. And this was the first, this and Zuckerkandl's evidence was the first molecular evidence that there was a close relationship between modern humans and chimpanzees. However, when you look into the literature, there was another person who investigated this who doesn't really get the credit that he deserves. And this is a guy called Albert Leighton, who used to be known as Albert Grunbaum, but who very sensibly changed his name during the First World War from Grunbaum to Leighton. And that's probably why his work is not as well known as it should be. In 1902, he wrote a letter to the journal called The Lancet, and it's called A Note on the Blood Relationship of Man and the Anthropoid Apes. And what he suggested in the letter, and we now know that basically what he was doing was a newfangled, what he was doing was using what was then a newfangled technique, which was mixing antiserum with serum, and that was called serology. And he wrote that I find that human antiserum gives a precipitate with all three species of anthropoid apes already named. And to me, this precipitate is practically indistinguishable from that obtained with human blood. In other words, if you mix human blood with human antiserum, or you mix human antiserum with the blood of these apes, and he got the blood from the zoo in Manchester. He was working in Liverpool at the time. And then he goes on to say, Occasionally, it is seen that the blood of the orang gave a more gelatinous precipitate, in other words, was more different as compared to the granular precipitate of the other bloods, in other words, the chimpanzee and the gorilla. But this may have been due to accidental circumstances. So he sort of said, you know, it may have been my fault. But basically, this is almost certainly the first use of serology to compare a modern human with a chimpanzee, a gorilla, and orang. And it is remarkable that Grunbaum, as he was then known, detected the difference between the reactions of the chimpanzee and the gorilla and the orangutan. He should get more credit than he has been given. And then, of course, Watson and Crick, with the unwitting help of Rosalind Franklin, once DNA was discovered, and you've heard from Mark Stoneking how much it can be used to explore human evolutionary history, more recent human evolutionary history, that confirmed the close relationship between modern humans and chimpanzees. And instead of the diagram I showed you at the beginning, we now know that modern humans and tanins, which is the family name, the tribe name for chimpanzees, likely share the common ancestor between 7 and 6 million years ago. And then a little more distantly come the gorillas, and then even more distantly come the orangs. So if we go back to our question, who are our closest living relatives? The answer is chimpanzees and bonobos. And then the other question, when do we share a recent common ancestor with them? The answer to that question is a little less precise. And some people argue that the common ancestor was older than 7 or 6 million years, and some people argue that it might have been a little younger. But I'm just going to use 7 to 6 million years as a reasonable interpretation of the evidence. So that's the first question. Second question, does our twig of the tree of life consist of just ancestors of modern humans, or do we also have close fossil relatives that did not make it onto the surface of the tree of life? Now, we have to have ancestors, but we don't have to have close relatives. I'm an only child. My father was an only child. So you don't have to have close relatives. This is a diagram of what is currently understood of the hominin fossil record. This is a pretty speciose interpretation, and the vertical axis is time. And each of these columns is a hypothesis about the existence of an extinct, or in the case of modern humans, we are still narrowly around, but the rest are extinct. The heights of the columns reflect the difference between the first appearance date at the bottom and the last appearance date at the top. As I said, this is a rather a speciose interpretation of human evolutionary history, but I think it's worth our consideration. The colors are an attempt to try and divide all the extinct ancestors or close relatives into grades. And the ones that are red are modern humans. The ones that are blue have a sort of a body plan, which is more like a modern human than like an ape. The ones that are the orange color and the brown color, these are extinct hominins that have very large postcanine or chewing teeth. And the green ones at the bottom are taxa that have been suggested that might be the first hominin, the earliest hominin. And then the light blue ones are hominins that are neither like the blue ones. In other words, they don't have a body plan, which is more like a modern human than an ape, but nor do they have the large teeth that are characteristic of the orange and the brown ones. Now this, in order to talk about the second question, I'm going to talk about not my evolutionary history as a member of Homo sapiens, but my evolutionary history as Bernard Wood. And so here is a picture of my great-grandparents' golden wedding. And this was a picture which was taken at my father's parents' home in Stoke Newington in North London in 1933. So this is my evolutionary history, not our evolutionary history. Now, when you look at that picture, on the left is my father's father. To his left, the one which is the guy with the spectacles, that's my father. To his left, to your right, as you're looking at it, is my father's mother. And then the two venerable people either side of the very exotic decoration are my grandmother's parents. So they are all my ancestors. They were necessary for my existence. The lady who is labeled with an R is a close relative. That's my Aunt Kitty. She was a delightful person, and I'm very pleased that I was related to her, but she wasn't necessary for my existence. And so in this picture, the ancestor to non-ancestor ratio is 5-1 in favor of ancestors. This is a picture of my christening party, which occurred at a date that I'm not going to share with you. And this was at my father's parents' house in North London. Now, when you look at here, there are some ancestors. There's my father's father on your left, and there's my mother, who is the woman with the A and the glasses. And then to the right, you will perhaps recognize my father's mother, and then my father's mother's mother from the golden wedding picture. So there are ancestors in that picture, but the bulk of people are either close relatives or my godparents, who are not related to me at all. So in that picture, the ancestor to non-ancestor ratio is much more equal. So let's go back to the diagram of the fossil hominins and inquire whether that is more like my great-grandparents' golden wedding or more like my christening. In other words, are all those columns, all those hypotheses about the existence of an extinct hominin, are they all our ancestors or nearly all our ancestors? Or are most of them close relatives and just a few of them our ancestors? And just to remind you, you must have ancestors, but you don't necessarily have close relatives. And when we look at those columns, you have to interpret the columns on the understanding that the columns are our present understanding of the earliest evidence for a fossil at the bottom and the most recent evidence at the top. It's racingly certain that we will probably find more ancient examples of each of those columns and more recent examples of each of those columns. And so those columns are going to expand in height. The more work people do to explore and look for new fossils. So basically, from just over 4.5 million years ago to very recently, there has always been more than one hominin somewhere in the world. And so if there's more than one hominin somewhere in the world, only one of those can be our ancestor. I mean, that's just the way it works. So, does our twig of the tree of life consist of just of the ancestors of modern humans? And the answer to that question is no. I'm sorry. Do we also have close fossil relatives that did not make it onto the surface of the tree? And the answer to that is very much yes. And it's more like an extreme version of the ratio at my christening than at the Golden Wedding celebration. In other words, most of the fossils that we recognize in the hominin fossil record are likely not to be our ancestors. So is it easy to tell close relatives from ancestors? Now, I've been very definitive about telling you which of the people in those images are my ancestors and my relatives, because I have evidence, which is, there is historical, there is written evidence. But in the absence of that historical evidence, how do you tell close relatives from ancestors? And the answer is it's rather difficult if you're trying to use the skeleton to answer that question. So in theory, reconstructing phylogeny should be pretty simple. The more morphology you share, the more evolutionary history you share. But there is a problem. And the problem was identified by this guy called Eric Lancaster in 1870, who was the director of the Natural History Museum. And he wrote, when identical or nearly similar forces or environments act on two or more parts of an organism, the resulting correspondence is called forth. And don't forget, he was a Victorian, so this is very flowery language. The resulting correspondence is called forth in the several parts of the two organisms will be nearly or exactly alike. In other words, two closely related organisms exposed to the same environmental challenges are likely to respond in similar ways. And he says, I propose to call this kind of agreement homoplasis or homoplasy. Homoplasis never caught on, and in fact, homoplasy didn't catch on for a long, long time. This was 1870, and basically his observation was buried, and nobody really took much notice of it until molecular evidence came along, which was able to give us much more secure information about relationships. And it was only when people realized that morphology sometimes clashed with the molecular evidence that people rediscovered homoplasy. So think of homoplasy as noise that obscures the phylogenetic signal. How do you identify homoplasy? That's another talk. So I would like to thank all my students and postdocs and my colleagues who've contributed to making me seem a much smarter scientist than I really am. And I want to reiterate my thanks to the organizers. And if there's time, I'm happy to answer questions. Thank you.
Thank you very much, Bernard, for the great talk. Hello, Craig. Can you hear me?
Yes.
به عنوان آخرین سخنرانی در خدمت دکتر کریگ استنفورد هستیم، استاد زیست شناسی و انسان شناسی دانشگاه ساوت کالیفرنیا که حوزه مطالعهشون بیشتر روی روابط رفتاری و اکولوژیکی نخستی‌های غیر انسان هست. خیلی لطف کردن و دعوت ما رو پذیرفتند و ما در خدمتشون هستیم. So we can start.
Thank you, Arash, and thank you for the invitation to speak, and it's really good to be here. I am a primatologist, and so I think my contribution to your conference these couple of days will be to give you a perspective on Darwin and Darwin's ideas about early human evolution from the study of wild great apes, especially wild chimpanzees, something I've been doing and my graduate students have been doing for a very long time. And my intention is to tell you where we are at this stage in our 100 years or so of study. I'm going to show you some of the work of my recent and not so recent graduate students. Let's start with our family tree. So you've heard a lot already about our phylogeny, and I will simply say that I always begin a talk like this by warning everyone listening that there is such a strong tendency to think of chimpanzees and bonobos, their close relatives, as under-evolved humans. Of course, we did not evolve from chimpanzees. We and chimpanzees have the same common ancestor that lived, as you heard, somewhere around 6 to 7 million years ago. And the lineages that you see there, depicted very simply, have diverged. And over the past 6 million years, we and chimpanzees have gone on into very different directions. So ironically, even though I used chimpanzees for most of my career to understand early hominin evolution, they really are not the best models for many aspects of human biology. For instance, even though we've experimented on them for a century or more, they make poor models for aspects of biomedicine. Chimpanzees do not get many of the most common cancers that humans do. Chimpanzee heart disease manifests very differently than in humans. So there are limits, but in general, I think we can use chimpanzees as a kind of a reality check to try to understand something about ourselves. So as you've heard already with Mark Stoneking's talk, Bernard Wood's talk, Don Johansson's talk yesterday, and you'll hear more tomorrow, and also all the talks in Persian, there are different approaches to the understanding of where we came from. Darwin knew of all of these. In some sense, he understood all of these, maybe except number 4 here. But of course, in Darwin's day, very, very few, practically no human fossils have been discovered, and they certainly were not well understood. He was fascinated by the orangutan in the London Zoo, so he understood that connection between ourselves and our close relatives. Of course, he didn't know what genes were, let alone DNA, so molecular genetics was not something he could begin to approach. And considering the lack of information in his day, he did an amazing job of trying to understand human origins that he published sort of in The Origin of Species, but not really, but then 12 years later in his second great book, The Descent of Man. So today I'm really talking about number 2. But it's really important to remember that the only actual physical direct evidence for where we came from is the fossils themselves. Everything else is in a certain sense circumstantial, even as important as it is. So genetically speaking, we know a great deal these days about the similarities and differences between humans and chimpanzees. I just put this slide up simply to say that there are these figures that are out there. I put 98.8% identical. That still leaves an enormous number of loci at which there are differences between us. The functional differences between humans and chimps and the other apes genetically is really what matters. So there are a number of important figures in our field, and you're hearing from some of my most esteemed colleagues in the world these couple of days. But I like to point out Jane Goodall. And if you don't know who Jane Goodall is, she is today an iconic global figure representing environmental issues and social issues, but she's also a pioneering primatologist. And this is a big year for Jane and for all of us who know and love Jane, because she is turning 90 years old on April 3rd, so just a few weeks from now. But I also always want to point out that as important an early researcher as Jane Goodall was, beginning in 1960 when she went to Africa and began what is probably the most important and certainly the longest study of animal behavior of any kind of animals ever conducted, and still continues today, that there are many other individuals. And one such individual who passed away a few years ago is my Japanese colleague, Toshisada Nishida. In general, the world does not know Nishida's name nearly as well as Goodall's name, and that's really unfortunate. It's partly because he didn't have the attention of the Western media for the decades that Jane has, but it's also because Jane Goodall was the pioneer who made many discoveries that I'll talk about in the next few minutes about chimpanzees. Nishida, 5 years later, he began his study 5 years later, only 100 kilometers from Goodall's study in Tanzania, in East Africa, and he confirmed all of the findings that Goodall had made. So you know the way science works is when you make the discovery, you are the person we all look to. And when you're the person who confirms those discoveries, which is really equally important, you don't get the front page. And so Nishida is a very important name to remember in the history of study of primates. All right, so the life cycle of a chimpanzee is strikingly similar to that of a human, up to a certain point. So let's compare females first. Here's just a very simple version of the life cycle of a female chimpanzee, born after a very similar length pregnancy to our own, several years of nursing, physical dependence, meaning if the mom dies, the nursing baby will die. There's very, very little adoption in chimpanzee communities in the wild. And then even after weaning, after the baby is no longer dependent on mother's milk, the baby is psychologically dependent on the mom for years after that. And if the mom dies during that time, even though the baby can feed itself, the juvenile can feed itself, the baby will still struggle to survive because there's not really a network of adoption in chimp society. A female chimpanzee goes through puberty, roughly age 11. That's quite similar to humans. First birth is typically the mid-teenage years. Again, similar, at least biologically similar to a human species. And then a baby about every 4 years until death. And I have there no menopause. Now there are some very recent data suggesting that menopause may exist in female chimpanzees based on some of the field studies. But I think we can say it's not really very similar to what we see in humans. There's no absolute cliff or ending to the reproductive cycling as we see in humans. And it's also important to point out that chimpanzees' reproductive system kind of develops over time during her life at a very similar rate to the rest of her body. Whereas in humans, our reproductive systems for women, again, stops at a much earlier stage of our lifespan. So female chimpanzees might live to be 40 in the wild and 60 in captivity, but they retain the ability to reproduce well into their 50s and even 60s, which is something that we don't see in humans. So there are these interesting differences that most likely have evolutionary meanings that we don't 100% understand yet. Meanwhile, when we talk about a male's life or a male's career, they're driven by different motivations. The motivation by both male and female is to successfully reproduce, of course, and successfully have offspring that grow up. But males have a very different approach to doing that. This is the same chimpanzee at 3 stages of his life, one of Jane Goodall's famous chimpanzees. Frodo, who is probably still the largest chimpanzee recorded weight-wise at Gombe, at Goodall's field site, and was an alpha male for a period of time. Here he is at age 18. He's a teenager. He's huge. He's physically powerful. He's not socially intelligent yet, just like most teenagers are not yet. And then in his 30s, he's beginning to look older, but he's a politically powerful animal. And then a photo taken by another researcher just before he died. He doesn't look like the same animal. He'd acquired some kind of a disease, never really diagnosed. And during this time, my point in showing you this slide is that he, of course, has the same genotype. His phenotype, his physical appearance changes dramatically, but his genotype is the same, and he's fathering offspring at all of these stages. So in terms of what drives female chimpanzee mating system, reproductive system, it's interesting just to note that when Frodo was even 12 and 14 years old, he was secretly fathering offspring with adult females in the community. So again, a career that we'd like to understand better. And when we talk about humans, this is a little conference paper that I did with colleagues a few years ago. you know, we have this idea, if we look at, for instance, well, any world leader, but US presidents, you see a photo when they took office, you see a photo when 8 years later they're done being president, and they always look much older. Their hair has gone gray, and we tend to think, oh, it's all about the stress of being president or being any world leader. It really ages you. But in fact, when we looked at the data, both for US presidents and for male chimpanzees, what we found is that males who become alpha or men who become president live significantly longer than those who don't. And there are a lot of different factors here. In the human case, of course, in early American history, many of the men who became president were from very wealthy families, even now today to a lesser extent. So they had benefits of medicine that others didn't have. But it's interesting because we think of being at the top as being very stressful, but there's obviously something very good about being at the top also in terms of male lifespan. This is something that one of my grad students did. We all focus on aggression and violence in primate societies. We try to understand human violence by understanding animal aggression and lethal aggression, which chimpanzees certainly engage in. So one of my graduate students, Jess Hartle, did a lovely study of reconciliation in wild chimpanzees. We know less about reconciliation, but it's equally important because an extremely high percentage of all the interactions that happen in chimpanzee society involve not hurting someone else, but either maintaining or reestablishing harmony in the community. And that's often the role of the alpha, is to maintain stability and harmony. But there has to be a mechanism for that. And this obviously applies to human societies as well. It applies to relationships in families, among friends, and between nations. There have to be mechanisms for maintaining some kind of harmony. So male chimpanzees are famous for being very aggressive. In fact, they're one of the few primates, other than humans, that engage in lethal territoriality. So here are a couple of photos taken by one of my former graduate students, Martin Muller. And these were taken at his research site in Uganda when he was doing his PhD. On the bottom, you see a male chimpanzee who wandered into an area that was a disputed area between the territories of two communities of chimpanzees. And in that area, he was essentially lost, it appears, and he was attacked by the males of the neighboring community, which were being studied, which Martin was following. And you can see the result. This male was killed. The close-up photos, which I would not want to show you, are very brutal. He was brutally killed. And all of the injuries were, chimpanzees don't have claws like a lion or a tiger. They have nails like ours. The injuries were all done through brutal aggression with teeth and also with hands, with fists. And then on the top side, just an interesting perspective on this. These are the organs of that chimpanzee that were done on necropsy, and you can see that obviously the one hand holds the brain of that chimpanzee, and another hand holds one of the two testicles of that male. And we often joke that these are the two organs that drive male chimpanzee behavior the most, and sometimes one takes precedence, and sometimes the other one takes precedence. Above all else, chimpanzees are amazing forest rangers. They have an amazing ability, because they're big brain, because they're smart, at understanding their environment. So this is taken in Gambia and Tanzania many years ago, and this fig tree is the only fig tree of that species that grows in the national park where the chimps live. And every September, it would put out massive quantities of fruit. The chimpanzees knew where it was. They would return to it year after year. They would eat every single fruit on the tree over a period of days. And then they would walk away somewhere, and their droppings, their feces, would contain the seeds of these figs. We would debate, and we still debate, the spatial memory of chimpanzees. Do they know where every single tree in the forest is? That would be thousands of trees. Or do they know where some trees are that are favored, that are needed, and then they locate other trees in the course of their travels? And I always would draw the analogy to if you go to a market once a week to do your shopping, and if that market were not the size of a normal market, but were instead several square kilometers in size, if you spent your entire life in that market, you would know where everything is. You would know exactly what season and what month. You would find certain fruits and certain vegetables because that's when they're available. And so chimpanzees may have that level of spatial memory, and that's something else that we find fascinating in terms of understanding, for instance, the way our ancestors used the landscapes that they lived on. So chimpanzees are primarily fruit eaters. This is something that is consistent across many different forests in Africa. In equatorial Africa, they also eat leaves, but an interesting small percentage of their diet you can see here, and this is based on the time spent feeding, and it's a very generic pie based on many studies averaged together. They also eat meat, meaning the muscle tissue of other vertebrate animals, particularly other primates. And they also consume honey because they love to raid bee nests, beehives. And they also eat insects such as termites and ants. This has been studied for many years, and it was actually Goodall herself who in 1960-61 discovered a chimpanzee using a stick to probe into a termite mound, these enormous earthen mounds that will hold 10 million or more termites. And these are very large termites. These are large insects. And she'll put that probe, which she has stripped of leaves, into the termite mound, and then she'll draw it out, and the termites will defend their nest by clinging to the stick with their jaws, and she will eat them all. And I had a grad student, I think I have a slide here, Rob O'Malley, who studied the nutritional ecology of this behavior and really did a wonderful job mapping the use of the mounds with GPS, understanding the spatial memory that chimpanzees have of where the mounds are located and the most direct routes between them, and also understanding the nutritional value of different kinds of termites, ants, and other insects that the chimps include in their diet. In general, these insects are a wonderful source of protein, fat, and carbohydrates. So we have many different kinds of tools that have been discovered. We can truly talk about chimpanzees as cultural animals, and really the realization that this wasn't just a random thing happening in some forests, but rather a systematic behavior across almost all the forests where chimpanzees live in Africa, really emerged only over the past 20 or so years as we began to pool the long-term data from many studies together. And now we can really say something about that. But here are some recent examples of chimp tool technologies that we did not know about in the early days of chimp research. Composite tools, meaning the chimps will use a big branch, a big stick to break open a termite mound or an ant nest, and then they'll switch to a smaller stick in order to gather the insects inside. Digging sticks, which are used in various places to, for instance, in the dry season when water is not available, when the streams may stop flowing, the chimps will use sticks to dig into the sand or the soil until they reach water under the ground so they can drink water out of it. Hunting spears, in quotes, because they're not really spears. The media called them spears, but my colleague Jill Preetz in Senegal has done a wonderful study approaching 20 years now, and she's documented chimpanzees there using branches and sticks to insert into holes in trees in order to either kill or injure small animals in those trees and then pull them out and eat them. So another protein source. So they're using a weapon in hunting, and that's really the only case in which chimps use tools as weapons. And then finally, chimpanzee archaeology, which fascinates people interested in human evolution, because we now know that in some forests in West Africa, there are aggregations, there are gatherings of stone tools that chimps have found in the forest and brought to a central place in order to use as hammers and crack open nuts. And then those tools have been left there. And over a period of time, thousands of years, they've become covered with layers of soil, so we can actually dig down into the soil and uncover these tools. So actual archaeology, material culture created by chimpanzees. Yeah, so here's the study by my student, Rob O'Malley. He really got a good handle on why chimps are eating these different animals. He also mapped the different termite mounds. He studied the insect side of this system as well as he studied the human side. Another grad student recently finished who did a genetic study. So in many parts of Africa, as Africa develops and human populations grow, chimpanzees are living in closer and closer proximity to people. And Maureen McCarthy did a lovely study using mitochondrial DNA of trying to understand the composition and the nature of chimpanzee communities living in areas where they were crop raiding. where they were invading farms to take food that people are growing out of the farms as part of their diet. And people are harassing the chimps. So the chimps are afraid of people. They can't really be watched or approached closely. So she collected chimp feces and did a study that, in fact, she worked in Max Planck, where Mark Stoneking is located, for her postdoc work to really understand what was going on there. So these are the kinds of things we can do these days, especially in a changing world in which humans and chimpanzees live closer and closer together. So my own work, done beginning long ago, which I'll just summarize now, was really about chimpanzee meat eating. And there was a long literature going back to the 1960s and Goodall's earliest observations of chimps as meat eaters. But it was always an issue of, well, chimpanzees eat meat, but so little that it really doesn't give us any kind of meaning of the earliest origin of hominin meat eating. And the fossil record is, of course, that about 3 million years ago, we see clear evidence of meat eating because we find cut marks made by ancient stone tools on ancient fossilized bones of the animals that were being eaten. It's hard to say whether the animals were killed or scavenged, but we have clear evidence of meat eating based on these stone tool cut marks. But before that, we don't have a signature in the archaeological record. So we turned to chimpanzees to try to get a window into what might have been going on. And that's really was the justification for what I did. This was my postdoc work initially long ago, and some of my students have followed this along. I have a graduate student right now preparing to go back to the field and continue kind of more sophisticated questions related to what I did back then. So first of all, we know that meat was an important part of the diet. We don't know how important. The signature is not clear. Did earliest humans eat meat once a week, once a month, once a decade? We don't know this. We don't know the answer to that. We know that animal protein and fat is in short supply in a forest. In most African forests, a chimp could probably find more fat in plant foods than in animal foods, and at much less cost in terms of calories expended hunting and trying to catch animals. Of course, the fat would probably not be saturated fat. It probably wouldn't have the same amino acid composition and so forth that animal protein, that meat would, but the protein and fat is there. It's in short supply in general if you're going to look for animal protein, and it has to be caught. And also, lastly, some of my work focused on what does the meat mean to the animals once it's been caught and killed. You see this as a chimp on the bottom. This is actually Freud, one of the early chimpanzees from Goodall's study at Gombe, who's tearing apart a colobus monkey, eating a colobus. And meat is obviously of nutritional value, but then it also takes on a role in chimp social systems in terms of being a tool for bartering, for securing an alliance, for snubbing a rival. And it becomes part of their politics. So that made it also an interesting topic to study. So at Gombe, there had been one brief study of hunting behavior done by Geza Teleki in the late 60s. He was a grad student at Penn State in the US at the time. But when I approached Goodall about doing this in the 1990s, no one had done anything very systematic on hunting. Now, today, in the 2020s, we have more studies that are longer term and more systematic than mine was. But at the time, it was the most detailed study. So what we learned through the study that I did and with students is that most hunting is done by males. It's really a male behavior. And when I say here that the behavior is partly female driven, I'll come back to this. What I mean is that female reproductive cycles and female swelling cycles, because female chimps develop these swellings on their rear ends when they're ovulating, these encourage males to be social because the males gather around the females because they all want to mate with her. And that leads to the males being in numbers, and that leads to the males hunting and being successful when they hunt. 2/3 of encounters result in hunts, but not all. One of my key questions was to try to understand when chimpanzees are walking along the forest floor and they encounter a tree holding colobus monkeys, that's the main food, why do they hunt sometimes but not other times? And that was something I devoted a lot of time to and lots of publications about. They're successful about half the time. That might not sound extremely high, but actually that success rate, that in half of all hunts, at least one prey animal, like a monkey, sometimes a pig, is killed, That's an extremely high success rate. That's higher than you would find for wolves. It's higher than you would find for lions or cheetahs or leopards or most carnivores. And those animals, of course, are carnivores. Their diet is 100% meat, whereas chimpanzees are more like us. They're omnivores, whose diet may contain some fraction of meat. And in addition to being successful half the time, they sometimes will make kills of many animals at the same time. So in my case, the maximum was 7 in a group of 30 monkeys. The chimpanzees attacked, the male chimpanzees attacked, and about 1/4 of the group of monkeys were actually killed. These are old data from my study showing that hunting is seasonal. And in most studies in Africa of chimpanzee hunting, the hunting is seasonal. Now, it happens that in Tanzania, where these data come from, there are 2 dry seasons, and the hunting peaks, and these are raw numbers of hunts, are in the dry season, in the 2 dry seasons, and particularly July, August, September. So that's interesting because the data that we have from some early hominin sites in the same region in East Africa suggests the same thing. And as to why that would be, there are lots of ideas about that. Some people initially thought, well, maybe there's less food available in the dry season, so the chimps hunt in order to have more protein in their diet, in order to just have meat. But in fact, when we began to do detailed vegetation studies, we learned that in fact, there's a lot of food available in the dry season. The leaves are not there, but in some forests, fruits are widely available. And so depending on the composition of nutrients that you're seeking, the chimps don't really need to hunt more in the dry season. So something else might be going on. And as it turned out, what we found was that this was related to, at Gombe at least, female reproductive cycles. Female chimpanzees do not give birth seasonally. They give birth at a relatively consistent rate in every month of the year, but their swellings, their cycles, their visual cycles in terms of those swellings that they get are seasonal. And that contributes to chimp hunting. We also found that meat consumption was very interestingly higher than we expected. So grams per day, now this doesn't look like a lot. If you were to buy a hamburger in a cafe somewhere, you would get a lot more grams of meat and of protein and fat just in that one hamburger than any of these are showing. But in terms of the average amount of meat being consumed by chimps during the peak hunting months, this is more than we expected. And so it meant that when we do comparisons of traditional human hunters, so-called hunter-gatherers or foragers, and we compare them to what modern chimps do, we find that chimps are not somewhere off the bottom of the graph, but rather somewhere in the lower third or so of the graph, depending on the comparison that we're making. And that was really unexpected. So just turning a little bit, another one of my former graduate students did a wonderful study trying to understand the role of hormones in chimpanzee behavior, in the behavior of male chimpanzees. We've known for a long time that testosterone plays a role in the social lives of human males and non-human animal males as well. If you, for instance, were to watch 2 men playing tennis, you find after the tennis match, although you might think that the winner of the match would experience a rise in testosterone because he won the match, in fact, what we really see is that the loser of the match experiences a drop in testosterone. So that's been demonstrated also in wild non-human primates. Robert Sapolsky at Stanford University was the first to really show that for wild baboons. And Martin Muller was the first person to show that that's also true for chimpanzees. This is really important because, we want to have a baseline of some kind for understanding that the mediation of aggression involving endocrine systems, involving hormones, and he really was able to begin to address that. The hardest part of the study was obtaining the samples. He used urine, and he relied on the fact that when male chimpanzees wake up in the morning, high in a tree where they've slept the night before, the first thing they do in the morning is what we all do, they urinate, and then he was able to catch that. This is actually the son of one of my colleagues, Richard Wrangham, years ago, catching some of the urine, and then that gave Martin his sample, and he used either liquid nitrogen to freeze it, or he used filter paper to be able to analyze the content of what was in it. And that at the time was quite pioneering work as well. All right, finally, just to do a comparison here, I mentioned hunter-gatherers, and I said in my first slide or two that they were one of the other ways that we try to understand our ancestors. So first of all, obviously these people in the small pockets of the world where they still exist, because they're persecuted everywhere they live, they've been persecuted. Whether we're talking about Native Americans or people in Africa, Asia, Latin America, wherever, these people are modern people. They are cognitively as the same as you or me, but their technologies are very simple and very traditional. And so we like to study them, anthropologists have for generations, to try to understand what earlier humans who had simpler technologies might have done, how they might have lived off the land. My comparisons have mostly been between chimpanzees and these people to try to understand the energetics of being a human hunter versus being a chimpanzee hunter. And so there are some differences that are really important here. So for instance, humans hunt larger animals. And if you have several hunters together, usually they're men, they are potentially hunting big animals, a giraffe or a zebra, whereas chimpanzees are always hunting small animals. They have never been recorded to kill anything larger than about 20 kilos, and usually much, much smaller than that, usually just a few kilos, like a monkey. Chimpanzees we don't believe they plan hunting at all. They hunt opportunistically, meaning they are looking for fruit, they're looking for leaves, and they may opportunistically encounter a group of monkeys in a tree and then hunt those monkeys and potentially catch one or more. Whereas humans, of course, set out in the morning, often with a plan. We're going to go this way because we think there's a herd of animals this way. And when they reach the animals, if they find them, then there's a coordination of the hunt also. And that's the last item on this list, that humans will coordinate their hunting either by gestures or by something vocal, yelling to each other, whistling to each other. And that is something chimpanzees do not do. Bipedal versus quadrupedal, the third point, chimpanzees walk quadrupedally. They, contrary to most people's images of a chimp climbing in the trees, they climb to find fruit, they climb to sleep, to make a nest, but most of their travel is on the ground, sometimes for kilometers every day. And humans walk bipedally, of course, and we know that bipedal walking is a more efficient way of getting around than quadrupedal walking of the kind, knuckle walking, that chimpanzees do. So if you're bipedal and you're an efficient walker, you probably have the ability to save the calories that chimps are spending. So you can walk much further, and maybe that allows you to plan hunting, to be able to go out and say, we're looking for meat today. And as I said earlier, chimpanzees, except for that one case that I mentioned in Senegal, do not use weapons when they're hunting, whereas humans routinely do, whether they're spears, bows and arrows, or whatever. And chimpanzee cooperation in hunting has been a disputed topic for a long time. I had arguments in the early days of my research because I never felt, based on my data, that the chimps were actually cooperating, and other researchers did. And I think the consensus today would be that the male chimps are acting together, but not cooperating actively with one another, that there's power in numbers, but that each male is out for his own intake of food and not really thinking about anything altruistically, and then the coordination that I mentioned. And so, yeah, finally, we then, with a colleague, Caleb Finch, who's a prominent authority on the origins of meat eating and also nutrition and the aging process, we did a paper long ago that looked at the aspects of meat eating in terms of human evolution. And we basically argued that it was the origin of bipedalism, and that there was even genetic evidence to suggest that when you add meat to the diet in a significant way, there's a downside. There may be a downside in terms of correlations with infectious disease. There may be a downside. Finch is an expert on dementia and Alzheimer's, and he believes that it's linked to diet, and that the origin of meat eating may have been linked to the onset of those diseases in the human species, but that we really dramatically changed our biological plan as a result of the addition of meat to the diet in a way that took off exponentially when humans began to walk bipedally and then began to use weapons in a systematic way. Okay, I don't know if I've exceeded my time or if I'm on time, but thank you for listening, and I hope you enjoyed.
Thank you very much for the great talk, and thank you again for accepting our invitation. خب من تشکر میکنم از همه عزیزانی که تا الان با ما بودند. روز دوم همایش‌های روز داروین ما هم به اتمام رسید. برای اطلاعات بیشتر و دیدن ویدیوهای این سخنرانی‌ها میتونید به 2 کانال تلگرامی مدرسه مجازی فلسفه تردید و خبرهای تکاملی سر بزنید. فردا از ساعت 7 ما روز آخر و روز سوم رو شروع خواهیم کرد و امیدوارم که شما رو فردا هم ببینیم. خیلی ممنون و متشکرم از همگی و خدا نگهدارتون.